The overall goal of this procedure is to create nanopores in artificial membranes for use in protein adsorption studies. This is accomplished by drilling nanopores in a nitride membrane using a stem. Then, the nanopore is loaded into a chamber and it is wet with an ionic solution so that fluid forms a bridge from one side of the nanopore to the other. To make the nanopore ready for experiments, a voltage is applied across it, and a current of ions can be observed to pass through it. The final step is adding protein to the nanopore chamber. Ultimately, observing long-lived blockades in the ionic current across the nanopore reveals the adsorption of proteins to the sides of the nanopore.
Hi, I'm Dave Niedzwiecki. I'm a graduate student in the Department of Physics at Syracuse University. Though this method can provide insight into the adsorption of proteins on inorganic surfaces, it can also be applied to other systems, such as protein-nucleic acid interactions, single-molecule force spectroscopy, and the study of molecules in confined spaces.
Begin by bringing up the stem to an acceleration voltage. Next, load an SPI silicon nitride window grid into the TEM sample holder. Clean the grid with oxygen plasma for 30 seconds to clear debris before loading the sample. Load the sample holder in the TEM and allow for the vacuum to pump down. Using the bright field TEM mode, find the nitride window, a bright square on the ronchogram. Make sure the TEM is aligned properly, then align and focus the sample. Now put the system